Assuming we have successfully fused the cells and we have these fusion products, what comes next? So selection is done in the HAT media, which stands for hypoxanthine, aminocterne, and thymidine. Something we need to know is that in order to proliferate and divide, cells need to make more DNA. And to make DNA, cells need to make nucleotides. Now there's two main pathways for nucleotide synthesis in cells. One is the salvage pathway, which uses an enzyme called HGPRTase, and the de novo synthesis pathway, which just uses basic precursors. Now, the salvage pathway uses an enzyme called HGPRTase, and the myeloma cells that we used do not have HGPRTase. And the nature of the HAT media is such that the aminocterne in the media is a competitive inhibitor of an enzyme called dihydrofolate reductase, which is necessary for de novo synthesis of nucleotides. So de novo synthesis does not take place in the HAT media, and the salvage pathway can take place. However, the myeloma cells that we used do not have HGPRTase. Now, in the isolation step, we should note that the only cells that have HGPRTase are the fused B cells, the hybridoma cells, and the unfused regular B cells that might be present. Now, if we look at this diagram, we can understand this quite clearly. The unfused B cells and the fused B cells are the only cells, only undesired cell types that have the HGPRTase enzymes. Therefore, they're theoretically able to grow in the media. However, they don't grow because they're not cancerous cells and they are not capable of indefinite growth. The myeloma cells, although they are capable of growing indefinitely, they do not grow because they do not have HGPRTase, and the de novo pathway is inhibited in the HAT media. So the only cell type ultimately that is able to grow and proliferate is the desired hybridoma cells, and we're able to isolate it using the HAT media.